Anatomy of the nervous system. Again, this is just a review here. We talked about two divisions, two general divisions: central nervous system, peripheral nervous system. Okay, central nervous system includes brain and spinal cord. Peripheral nervous system includes our peripheral nerves and some of our cranial nerves. Okay, some of our cranial nerves. Now, in terms of the flow of communication, we talked about sensory information moving in this general direction from the world around us. Okay, it was going to be processed at centers in the brain. What do we call those centers? Integrating centers, correct, integrating centers. These are usually pons or medulla. Okay, and then we know that there's another flow of communication based upon this analysis in this general direction. Okay, and this can be either motor or autonomic. Okay, I'll talk about that in a minute. So basically here we have sensory, let's say sensory receptors, picking up sensory information from the world around us. There is an afferent signal. So this flow of communication is afferent or sensory. Okay, whereas this flow of communication is efferent. Okay, and this can be either motor or autonomic. Okay, and so we send out a signal to some type of effector tissue here, or some type of target cell. Okay, and the effect can be one of two types. Now, this is what we're going to speak about coming up next. So this is kind of a preview here. This can be an autonomic function or a somatic function. Okay, are two types of effector responses. And I'll just say here, autonomic is involuntary, means we don't control it, whereas somatic is voluntary. Okay, to be continued. So we'll continue talking about autonomic nervous system and then somatic nervous system, but it's really important for you to understand where it fits in everything we've been saying so far. Okay, so you take information up about the world around us, right? I'll just put here the world around us. Send that to the brain, specifically the integrating centers, that's back in the CNS. From the CNS, we send out an effector signal on an efferent type of fiber. So these are two different types of fibers, sensory fibers versus efferent fibers, okay? And then the signal that we send back can be a motor signal to run, to jump, to scream, to move, or an autonomic signal to raise blood pressure, to secrete glandular secretions, to increase heart rate, things like that, okay? All right, so that's the anatomy of what our nervous system looks like. Any questions on this? Any questions on that? So that was kind of a review. So now I'll go on to the specific review topics I just mentioned. Actually, I will draw this out. Okay, so I'll review the postsynaptic potentials here. And this is differentiating IPSP from EPSP. And so I'll just sort of review those here. So these are called postsynaptic potentials. We want to bear in mind that these are types of graded potentials. Okay, so don't let the fancy name fool you. Okay, and so we have two general types. There are EPSPs and IPSP. Okay, we know EPSP is going to be a depolarizing event to the membrane potential. Right, so the membrane potential is going to depolarize. What type of channels did we say was responsible for this type of event? Sodium or, not potassium, sodium or calcium, sodium or calcium, okay? And that means that we're going to open up a sodium or calcium channel. These are positively charged ions. They rush into the cell. This is a depolarizing event, okay? It's not an action potential. It's a graded potential, but it's still a depolarizing event. It could bring us closer to an action potential. As far as inhibitory or IPSPs, right, these are going to hyperpolarize the membrane potential. And what type of channels did we say are responsible for these events? Potassium is one, and potassium and chloride, potassium and chloride, okay? And we spoke extensively about chloride and its role in stabilizing the membrane potential, so make sure you review that. It's not only hyperpolarizing, but it has a stabilizing function as well. Okay, so these are EPSPs versus IPSPs. Let's look at our scenario with two neurons here. Okay, we spoke about this region of the neuron being what's important here. All right, so let's sort of label this out first. A presynaptic neuron, a postsynaptic neuron. Okay, and so our action potential is moving from this way. It gets to the terminal bouton. We spoke about all the events that happen here. Okay, that synaptic transmission. Make sure you review that. I'm not going to go over it here. And then what happens here at this axon hillock in the second neuron? Exactly, that's where we sort of analyze all of the graded potentials that have come in here. So we can have IPSPs, we can have EPSPs, we can have IPSPs, which are negative, and all of that is graded at this axon hillock, okay? This is the function that we call, I'm kind of writing this all in one color, sorry. This is what we call neural integration. We spoke about several different steps of neural integration. There was summation, right? There was convergence and divergence, right? Those different signals that are coming in or going out. There was also postsynaptic modulation, right? Which included synaptic facilitation, synaptic inhibition. So all of that is sort of analyzed here, and understand that we're analyzing these two types of potentials. IPSPs are coming in. IPSPs open up chloride and potassium channels. They hyperpolarize the membrane potential here. EPSPs are coming in. They do the opposite. They depolarize the membrane potential. And so at the axon hillock, this is a time for us to really consider everything that's coming in, negative, positive, and figure out whether or not we have gotten to threshold. If we've gotten to threshold here, we propagate an action potential this way, okay? And so we really want to associate this with the different types of channels as well. Along the axon, we find what type of channels? What type of channels are along the axon specifically? So we know they're voltage gated because they help with that action potential. Voltage gated sodium, voltage gated potassium. What type of channels are found on the terminal, the axon terminal? Voltage gated calcium. What type of channels do we see on the soma and cell membrane? What's happening on the soma? So on the soma, we're picking up signals, we're receiving signals. What type of channels did we talk about were responsible for picking up those signals? Either in a fast way or a slow way. We talked about ligand-gated channels, right? Ligand-gated channels. Those ionotropic receptors are ligand-gated channels. The receptors are channels. And when we bind that neurotransmitter, it's going to open up a channel, right? Whether it be sodium, potassium, depending on what it is, okay? So make sure that you're connecting these. The channels that we're speaking about here are the ligand-gated channels, okay? They're on this part of the membrane. And this is how we can process an IPSP or an EPSP. Okay, so make sure that you're making that distinction. So if it's a ligand-gated sodium channel, let's say the neurotransmitter that it's associated with opens up, that neurotransmitter opens up a sodium channel, that's an EPSP. If it's a ligand-gated potassium channel and that neurotransmitter that was released here binds to that ligand-gated potassium channel, that's an IPSP, okay? Opening up that channel is an IPSP, that's a hyperpolarizing event. So make sure that you're making those connections between different things we've talked about and distinguishing IPSPs, EPSPs, and then action potentials, okay? Any questions on that? What do you mean? Right, yes, that is correct. So if sodium channels close, that becomes an IPSP, whereas if potassium channels close, that becomes an EPSP. Okay, so it's going to be acting in the reverse here. Sodium is a little bit different because sodium can also have a stabilizing effect, just like chloride, depending on what type of cell we're looking at. If it's an excitable cell where there are active pumps on the membrane, or if there are no active pumps. But in general, the general idea is if you close a potassium channel, you will depolarize. If you open a potassium channel, you'll hyperpolarize. Okay, so just think about the ions that are moving, the charge on those ions, and the direction that they will be moving or not moving in if we close the channel. Okay? Any other questions? Any other questions? Okay, so the remaining things I'll review are briefly refractory periods, and I'll sort of just go over the slides for that, unless there are specific questions on refractory periods. And then the synaptic receptors. Okay, so when we spoke about refractory periods, we talked about the two main types, the absolute, which is basically during the duration of the action potential. And what was responsible for the absolute refractory period? What event or what channel? The closing of the sodium channel, specifically the inactivation gate on the sodium channel. And so we use the word inactivated here to really distinguish it from the closing of the activation gates. The inactivation gates close, the channel closes, okay? And so we physically cannot fire an action potential here, all right? If we can't open up that voltage-gated sodium channel, we can't depolarize the membrane, we cannot fire an action potential. And this is during the duration of the action potential, that 1 to 2 milliseconds, that is the action potential. Now, immediately after, right, or immediately during this hyperpolarizing part, we can fire an action potential because our inactivation gates are open, okay? So the channels are closed, and here I'm saying closed and not inactivated because the activation gates are closed, but the inactivation gates are open. So they are closed, but they are capable of opening. And so during this time, we could fire an action potential, but it becomes really hard because potassium is still at movement, right? Potassium is still moving. Potassium is still moving out of the cell. And doing so drops the membrane potential much more negative than rest. And so in order to depolarize a cell, we need to get the membrane potential to negative 55. And getting to negative 55 from negative 85 is much harder than getting to negative 55 from negative 70. And so we need a stronger stimulus to get to threshold during hyperpolarization, okay? Because the membrane potential is very negative. And simply because potassium is still moving, alrighty? Yeah, so I think that's the crux of the absolute and the relative. Okay, the why behind it as well, what ions are responsible for these periods. Any specific questions again on this? So this bit? Yeah, the sodium channels are open here. That's how we get this potential change. Sodium channels are opened, sodium's coming in, we're approaching sodium's equilibrium potential, which is very positive, but at that peak, at positive 30, we know that that signal was sent, right? When the negative 55 threshold was met, when this negative 55 threshold was met, 2 signals were sent here. One is to open the inactivation gate. So think about, we get to negative 55, 2 signals are sent. Open the inactivation gate, which essentially opens the channel, right? And then after 1 millisecond, close that inactivation gate, which essentially closes the channel. And at that same time, potassium channels open up. Okay, so let's go back to this slide. The activation gates close when we get back to rest. Okay, that's why it's really important to talk about the 3 configurations of the channel, because it gets confusing. So what voltage determines what the channel is doing? At negative 70, the channel is closed and capable of opening, right? Because the activation gate is closed, but the inactivation gate is open. Right, so let's sort of start here. So this is at rest. Activation gate closed, inactivation gate open. The channel is closed, right? And I'm going to differentiate that from being inactivated, and I'm going to say it's closed, but it's capable of opening here, okay? And this happens at negative 70. From negative 70 to negative 55, right, we have graded potentials, and if we get to negative 55, 2 signals are sent. Open the inactivation gate for 1 millisecond. After 1 millisecond, close it again. Okay? And so that really times or syncopates these events so that potassium channels open exactly when the inactivation gates here close again. So think about that negative 55 value. Think about those 2 signals being sent to open up inactivation gates for 1 millisecond only. And then after that 1 millisecond, inactivation gates closed. Potassium, meanwhile, on the other hand, let's go to this slide, also has that timed event, that timed signal. So at negative 55, potassium has a 1 millisecond delay to open its channel. And so after that 1 millisecond, after that negative 55 threshold, potassium channels open up, sodium channels close. And this happens at the exact same time. This is really important. This is why these are timed and the inactivation gates are timed. Okay, did that answer your question? Well, we can't fire another action potential while we're firing an action potential, right? So on this same cell, let's go back to this slide. I hope I'm understanding your question. So you're asking why we can't fire another action potential here? Why this is a refractory period? Yep, so the inactivation gates are closed here. So while we're firing the action potential, inactivation gates are open, right? They're open for this duration. And I hope I'm not confusing you further. They're open for this duration, but here they close. And so the absolute refractory period is all of this period while we're firing one action potential, but then also the remaining period here where we cannot fire another one. Okay? I hope that makes sense. Question? Sure. So at negative 55, there are 2 timed signals. The first timed signal is for voltage-gated sodium channels, specifically the inactivation gates on those channels. And so the signal is sent to open them up here, okay? And then after 1 millisecond to close them, alrighty? And so after 1 millisecond, those voltage-gated sodium channels, the inactivation gate closes. As they close, potassium channels will open up, the voltage-gated potassium channels. And that facilitates the refractory period here, the repolarization here. Okay, any other questions? Now, the relative refractory period, as I mentioned, is because potassium channels are still closing. So here the membrane has gotten back to rest, the sodium channels have gotten back to their original configuration where the activation gates are closed, but the inactivation gates are open. And so we could fire an action potential, but it's much harder, okay? All right. Any other questions on that, on the refractory periods? Any other questions? Okay. Lastly, I'm going to talk about those receptors, the synaptic receptors, fast and slow. here, and I'll summarize them on this slide. Okay, so this is basically comparing the fast response, which happens at ionotropic receptors. Those are ligand-gated, right? Ligand-gated channels, right? Please make that connection. And then metabotropic receptors, which are also ligand-gated, but they're not channels, okay? These are G protein coupled receptors. So they're connected to this protein, which brings about their response. So let's look at the difference here between these two processes. Now, the first one is a very simple event. A ligand, which is a neurotransmitter, that's released from our presynaptic neuron. So make sure that you're connecting the neuron part of this, where along the neuron this is taking place. So a neurotransmitter is released at the presynaptic axon terminal. That neurotransmitter crosses the cleft and binds to this receptor. Binding to this receptor, if it's an ionotropic receptor, opens up this channel, okay? Depending on what channel it is and what type of movement, right, as we correctly pointed out, whether or not the channel is closed or open, the charge on the ion in question will determine whether we hyperpolarize this cell and create an EPSP, or whether we hyperpolarize the cell and create an IPSP, or whether we depolarize the cell and create an EPSP, okay? Whereas the metabotropic receptors are much slower in their response. It's not that simple, okay? So the same event happens, a neurotransmitter is released, selectively binds to this receptor. Because this is a metabotropic receptor, it's going to stimulate this G protein, dissociate one of its subunits off, and that's going to activate another enzyme downstream. Now, the other enzyme is going to release some molecules, which we call second messenger, and that molecule is going to bring about some cellular response, okay? It's either going to turn on another channel that could hyperpolarize or depolarize, or it can activate proteins, it can go to the nucleus, it can release calcium, a whole host of different things that will eventually bring about this cellular response, okay? So it's much slower, requires many more players in the middle, but it's a much more amplified